All right. So one of the organelles that we talked about with the chapter on cell organelles was the centrosome and centrioles. We said, hey, we're going to come back to these a little bit later on. The centrosome is this organizing location within the cell that's going to organize microtubules or the spindle fibers that are going to attach to the kinetochores of chromosomes. These are just the assembly or the assembly line kind of to make the kind of the structures to help organize the chromosomes. So they're going to be almost like these little fingers that reach out into the cell, grab the chromosomes, and pull them to one side of the cell. There are two barrel-shaped organelles called centrioles within the centrosome. They are going to help assemble those spindle fibers a little bit more specifically. But again, they're going to anchor them. You can see here, anchor them down at the cytoskeleton and the membrane, so it can pull the chromosomes to one side of the cell. Now, these are at a right angle, and these are a kind of newer addition to our understanding. We're still trying to figure these things out a little bit, why they organize themselves into right angles. But you're going to see centrioles and the microtubules in this chapter a good bit. So just understand they're organizers for microtubules, which are going to reach out, grab the chromosomes, and pull them back. All right, so 9.3 is going to look at mitosis more specifically and cytokinesis within M phase. Our first phase that we're going to talk about is prophase. This is where the chromatin are going to start to condense and form those chromosomes. So think prophase and condensing. This is also where the nuclear membrane, you can see around here, the nuclear membrane starts to disintegrate. We have to assemble and then organize these chromosomes and then pull them apart because these chromosomes have 2 chromatid per chromosome, and we need to split that genetic information so the new cell is going to have enough genetic information to survive. Now, I add in here these asters that are the microtubules that come out of the centrioles. We don't really have to understand that they make the star pattern, but I'm just showing you here that they are going to reach out and be the structures that actually pull these mitochondria apart. Or not the not the mitochondria, the chromosomes apart. I'm just showing you that these again, this doesn't happen just randomly. There is a lot of networking and a lot of communication and protein production here. So this is a very complicated whole process. Our next phase, which I don't teach in biology, so I'm actually teaching this stuff in my biology classes. This is a step that I really don't teach. I just let this, I kind of go from prophase to metaphase. So this is kind of more AP stuff. So with prometaphase, you're going to have these kinetochore regions on the outside of the chromosome, specifically around the centromere. The spindle fibers are going to attach to these kinetochore protein regions, and this is just an extra step here, like I said, that AP has to understand. Now, again, chromosomes aren't oriented yet. What that means is they're not on this metaphase plate. They're just attaching here. I have them attaching and being on the metaphase plate at one step in biology. Here, we're going to add in this extra step. They attach at this step. I like to describe this as those Chinese finger traps. The kinetochores and the microtubules are going to interwound their specific parts here, these different protein parts, and that's how they're going to split apart. This is really cool looking here. In metaphase, we're going to have the chromosomes being aligned along that metaphase plate, which is just in the middle. So they're just along this one line. This is really easy to identify in the cells. You can identify this by just the straight line of chromosomes. Now, there is a checkpoint here. We're going to come back to checkpoints a little bit later on, but there is a checkpoint to making sure all of the chromosomes align properly and we have the right amount of DNA. If this doesn't occur, the cell will try to fix itself by fixing the amount of DNA or fixing the orientation of the chromosomes. However, if it doesn't happen, the cell will go through something called apoptosis, which we'll get into, I think, 9.5. But this is where the cell has a checkpoint to make sure everything's working right. If everything's not working right, it will stop the cell division. You can again see this is the line of chromosomes that we're talking about. We have the centrioles right here coming out, and they're going to attach to those chromosomes at the kinetochore, which is around the centromere. There's a lot of verbiage here, a lot of words you got to remember. So just try your best here. Our next step is anaphase. I always relate this. I remember my first year teaching, I had a girl named Anna, and I said, hey, this is Anna. This is where, you break up with your boyfriend. Your chromosomes are going to break up with, or your chromatids are going to break up within your chromosome. Think about this as like Anna breaking up with her boyfriend. So the sister chromatids are going to be split apart by the spindle fiber shortening, and this is the shortest phase because it goes very quickly. So again, the chromosomes start migrating to the opposite poles of the cell toward the centrioles here, and they're going to be split apart. So again, this is technically still a diploid cell, but we're kind of doubling the amount of chromosomes because before, if you look, if we can go backwards, before, if you look here, our cells had 4 chromosomes with 2 chromatids. Now our cells have kind of 8 chromatids, but only 1 chromatid apiece. But it's still technically diploid here. All right, let's just skip this for now. Da-da-da. All right, telophase. Telophase, the chromosomes are going to start to uncondense because they have enough genetic information on both portions of the cell. You can see this cleavage furrow, which is going to be where the cytoplasm starts to split, which is going to be the next step. So again, you can see this the nucleolus also start to reform. This is where the cell is saying like, hey, I have enough genetic information. We've done everything. We're going to start to divide now for the complete mitosis. Let's see. Anything else I want to talk about? No. I will go over this chart in class with you. Let me go back. I will go more in depth with this chart in class, but understand here, we're doubling the amount of chromatids at a certain point, and then we're going to divide them in 2. So our resulting cell has 46 chromosomes with 46 chromatids apiece. We will double the number of chromatids after S phase, though. All right, and then again, within the M phase, we have cytokinesis, which is just the division of the cytoplasm. It's really hard to tell the difference between telophase and cytokinesis, which we're going to get to next week by identifying cells. But this is where the cell starts to divide the cytoplasm. So I won't be very technical when I ask you to identify the different stages here between telophase and cytokinesis because they are so similar, and it's really hard to tell the difference. So one little difference between the mitosis in the animal cells and plant cells. In the animal cells, you have this microfilament ring that helps split the cell. This is just a protein ring that's going to form, and it's going to help, again, just divide that cell. In the plant cell, it's going to be a little bit tougher because you have to have the cell wall between the 2 cells. So what happens is you start having the cell plate where cell wall starts to form, and after the cell division occurs, and again you can see the Golgi body here set in those little vesicles here with cell wall material. It's going to split between these 2 cells here, so it's a little bit different between plant animal cells, but just understand that there are differences, and these differences have evolved over time. I'm not going to show. I'll probably show you these videos